estimating densities of the cougar is highly important for their management and conservation. This is why the increased use of conservation detection dogs are useful in identifying individual animals from DNA extracted within their scat. These dogs are trained with similar techniques to those of bomb, drug, and search and rescue work, and these are able to locate and distinguish scat from the target species over large distances while ignoring any scat from species that are non-target. The first objective of this study was to assess efficacy of the use of scat detection dogs to locate cougar scat and obtain DNA of individual animals. And the second objective was to then use 4 abundance estimators to have multiple forms of results to compare. These estimators are the Huggins closed population capture-recapture, Markov decision process, CapWire, and a modified spatially explicit capture-recapture model. This study was carried out from the 8th of June to the 4th of July in 2011, within a 220 km² section of the Mount Emily Wildlife Unit in Northeast Oregon, where a concurrent cougar predation study was also occurring with 4 cougars being captured. They were then fitted with GPS collars and their DNA information was recorded before being released back to compare these to individuals identified from the scat samples to assess the efficiency of the methods being taken. Of the 251 scat samples that were sent to the lab after the 18-day study, 173 were cougar, suggesting a 68.9% success rate. 23 were bobcat, 17 non-target species, and 38 of them were unable to amplify. There was also at least 1 scat sample from each of the 4 GPS-collared cougars found in the samples. This table is a summary of the number of scat samples collected and the number of individual cougars that were identified from these out of the 4 study sessions carried out. It can be seen out of the 251 scat samples that 73 revealed individual identification, 41 unique individuals were identified out of those 73 captures, and 21 new individual cougars were found across the 4 study sessions. The 4 estimators were then used to provide density estimates of cougars per 100 km², with 95% confidence intervals. As can be seen, the average total density is around 4.5 cougars per 100 km², with the probability of a dog being able to find a cougar scat on this landscape within any of the 4 surveys calculated to be around 0.99. The reported capture probabilities in this study were the largest observed out of any previous studies conducted with wild felines, which further demonstrates the benefits of scat-detecting conservation dogs used to estimate cougar densities. Although the sampling area was relatively small, the study was able to show that estimating cougar populations with the use of conservation detection dogs is feasible, and could easily estimate populations on a broader scale if additional survey effort was utilized. Survey time and effort is also reduced, which makes it more affordable for more frequent and further expanded monitoring. Management of cougars is regularly questioned, and so estimates that are highly accurate and precise will ensure that the managers are able to make informed decisions when it comes to cougar management, particularly within Northeast Oregon.